to today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom. We'd like to thank our episode sponsor, Takeda Oncology, for their support of Myeloma Crowd Radio. And we'd like to celebrate today because today is our 100th show. Over the last 3 years, I've been privileged to interview myeloma experts on such a wide variety of topics and from investigators all around the world. I'm thrilled to help share their truly amazing work. And we're very pleased today to have some researchers from Germany join us. Our show today, we know that you've heard about monoclonal antibodies, which target a specific protein on the surface of myeloma cells. This is a very unique approach, targeting both a protein on the surface of myeloma cells and at the same time an activation of T cells. And we have with us today Dr. Hose and Dr. Seckinger from the University of Heidelberg in Germany. So welcome, doctors.
So good morning to America from Germany. And of course, it's a very big honor for us that we are with you at your 100th birthday. So I would like also to welcome you on behalf of my second in command, Anja Seckinger.
Hello, also from my side.
And myself, and we are very honored to give this talk and this discussion today because we think it's a very important point to discuss and interact also findings in research and to work together, we as clinicians and researchers with our patients to finally get a hold on a cure of myeloma. So Jenny, thank you for the nice introduction.
Oh, well, thank you. Let me introduce you actually both before we get started because we have a lot to talk about. Dr. Dirk Hose is head of multiple myeloma research laboratory at the University of Heidelberg. He's a practicing physician at the University of Heidelberg in the Department of Hematology, Oncology, and Rheumatology. He was awarded the Young Investigator Award at the International Myeloma Workshop in Paris and was granted the Ludwig Strauss Habilitation Award for Excellence in Research. And I found it most interesting that your doctoral thesis was on the topic of asymptomatic multiple myeloma, molecular background of progression, evolution, and prognosis, which I think is one of the most complex topics about myeloma when you're asymptomatic. Now, Dr. Anja Seckinger is deputy head of the Multiple Myeloma Research Laboratory at the University of Heidelberg in Germany and practices in the Multiple Myeloma Section and Department of Internal Medicine of Hematology, Oncology, and Rheumatology. She's been significantly involved with myeloma research in Germany as well as overseas and has received awards including the Young Investigator Grant at the International Myeloma Workshop in Paris, Abstract Achievement Award at the 2015 ASH Conference, and the Best Abstract Award in 2016 at the annual meeting of the German, Austrian, and Swiss Association of Hematology and Medical Oncology in Leipzig, Germany. So, doctors, welcome to you both. We'll probably just start with just a broad overview. So, and I know you can compare and contrast these with the monoclonal antibodies that patients may already be aware of, but what are these T cell bispecific antibodies, and in general, how do they work?
Well, Jenny, thank you again. So this is a very interesting question. So you have to think of these antibodies as something with two arms, which basically couples T cells by binding on a structure which is at the surface of the T cell, which is called CD3, and with another arm on another structure which is on a myeloma cell, which is called, in this example, BCMA, so it's B-cell maturation antigen. So basically the idea is pretty simple. These antibodies have two arms and bring just together myeloma cells and T cells. And now T cells are the most active, let's say, killers in the immune system. So they are very active in destroying tumor cells and immune cells, but usually in myeloma patients they do not fulfill their work because they are hindered. You can somehow compare this to T cells being a kind of savage dogs, but they have a muzzle. And now the T cells brings the myeloma cell and this dog together, and then it bites let's say, the burglar in our example, the myeloma cells. So it activates it, and then the myeloma cells are killed, and so you utilize the most active cell being able to attack myeloma cells, which is actually the T cell, to attack the myeloma cells and destroy them.
Mm-hmm. And the difference would be between, because I know patients are familiar with daratumumab and elotuzumab, which are monoclonal antibodies, and they activate the immune system in a certain way, but differently than the T cells. Can you explain the difference?
Yeah, with a monoclonal antibody like daratumumab, this antibody only binds to myeloma cells, and then immune proteins, for example, are activated and produce a hole in the myeloma cell, which is then subsequently killed, or other cells which are less specific from the immune system bind to the cell and attack it. T cells are the much more potent variant of this, and the difference is that this T cell bispecific antibody has two binding sites, so the possibility to simultaneously bind the myeloma cell and the T cells and just bring them together, which is the difference in the mechanism of action. In principle, the molecule itself is quite similar. There are some interesting differences we will talk later on, but in principle, the idea behind it is very simple, of this just grabbing with one hand myeloma cells and with the other T cell, and this then activates the T cell for killing myeloma cells.
That's just a very visual description. We like to picture that in our minds of having T cells go after the myeloma cells and be joined. So when you say they're binding, you're binding the myeloma cell to the T cell, and then how strong is that binding, or how does that
so
work?
This would be an interesting question, because this is something for Anja, and I will just pass over.
Okay.
So this is indeed an interesting question, which was actually raised by Dirk, because as you might know, he's not only a physician but also a physicist. And at one point he asked, so what is the binding strength between a myeloma cell and a T cell? And finally we applied an interesting method called atomic force microscopy, which is able to measure the binding strength between, in this case, a myeloma cell and a T cell at a very sensitive level. And we did so in the presence of the antibody and without. And what came out is that in case the antibody is present, the binding between a myeloma cell and the T cell is increased by a factor of about 3 to 4. And the interesting thing is that when you look at the data, the units are in piconewton, and at least for us, this was not really quite familiar, because it sounds not that much. And we calculated what this means if you have a myeloma cell, and it's interestingly, it's 200 times the weight of a myeloma cell that is necessary to break the binding between the myeloma cell and the T cell. And when you think of yourself and somebody is tearing at your arm with 200 times your own body weight, this is then pretty much. So it's quite strong binding that we see between the myeloma cell and the T cell that is caused by the drug.
Yeah, very strong binding. Well, let's start talking about these two different arms, because you explained very well how it goes after two different targets at the same time, or to do the binding process. So first question is, I know some of these other monoclonal antibodies are going after different targets like CD38 or CS1, and this one is going after BCMA. So first, why BCMA as a target, and then we'll talk about the T cell arm a little bit.
Okay. So BCMA is called B-cell maturation antigen, and this basically describes at what time during the development of plasma cells from their early precursors is expressed. So it's very late expressed on plasma cells, and it's expressed on normal plasma cells and also on malignant plasma cells, so myeloma cells. And the very important point is that it's almost exclusively expressed on normal and malignant plasma cells. Now, and this is the reason why we choose it. Now, why is this important? This is important because I gave you the example with this savage dog before, and the problem is to control this dog. You do not want, in our example, this to attack bystanders, or in our patients that the T cells are falsely activated and then cause side effects because other cell types might also be bound by this antibody. So this is the important point, how to control the T cells only to attack the myeloma cells. And this is done because BCMA is very specifically expressed on myeloma cells and on normal plasma cells. So these are the only cells which are attacked. And this is quite a unique pattern because for other antigens, like you mentioned, CS1 or CD38, the spectrum of cells expressing it's much more broad, and therefore not only the myeloma cells as target cells are killed, but it also causes some of the side effects. So it's this pattern of expression limited to myeloma cells and normal plasma cells, but also it's reasonably strong expressed on the myeloma cells. You can easily think that this is important because when you think of this grabbing actually the myeloma cells with the antibody, when there are too few handles on the cell, you might go astray by trying to grab it, which is something which is in place for some of the antibodies or mechanism of resistance we might talk about later on. So this is the basic choice, and we screened a high number of potential targets, and this was actually from this expression the best target we have seen or identified. Mm-hmm. So this is the arm, why it's this specific arm binding to myeloma cells. And now for the T-cell arm, this binds CD3, and this is an antigen which is expressed on almost all T cells. And these T cells are bound, just selected as binding as described before, and brought together to the myeloma cells. And by this process of bringing the myeloma cell and the T cell together, the T cell is activated, and this activation is like it is also being done during a normal immune reaction. So this CD3 is one of the main T cell receptors, so a structure which helps the cell to communicate from the outside to the inside and to mediate this process of the activation.
Mm-hmm. And it sounds like it's different than, there are a lot of different targets, protein targets on the surface of cells, and you hear a lot of different targets in myeloma, but it sounds like that CD3 is like the primary target for T cells. Is that an accurate way of saying it?
Yeah, it's actually you can just imagine why I choose CD3, because it's expressed on almost all T cells, and so it's a very good target just to select almost all of the T cells. There are other antigens of T cells like CD4 or CD8, but they are always only expressed in a subfraction. Now, if you want to get more or less all T cells activated, then you have to choose something which is almost on all T cells, and it's as easy as that this is the case for CD3.
Mm-hmm. So you're enlisting more T cell help basically by going after CD3. So how long does it take for a T cell to kill a myeloma cell?
Well, the killing of a myeloma cell takes about 6 to 8 hours. You can calculate this when you just watch in a live microscopy, and you just can see myeloma cells being killed by T cells, and you can then just calculate the time of how long this takes, and this is about 6 to 8 hours. And then T cells, which are activated like this, are kind of serial killers, so they can not only kill one myeloma cell, but they are able to kill one myeloma cell after another, and they even proliferate, so they even multiply themselves when they are exposed with this antibody, so you amplify the reaction against the myeloma cells.
Oh, that's great that it's not a one-to-one. It's just continually expanded. So some more questions about the BCMA target. So how would you, I know there are a lot of myeloma cells. I had a researcher talk about how he was testing for BCMA levels, and the BCMA would go up and down, and it would almost correlate with the M spike. And I don't know if that's accurate or not, but he was working on a test to look at that. So if there is some BCMA, but not a lot, on normal cells, how do you avoid it binding to normal cells with BCMA versus just myeloma cells with BCMA?
Well, the only normal cell which actually expresses BCMA is a normal plasma cell, and these cells are likewise targeted by the antibody, so they are also removed. But this can be quite easily coped, because what is not removed is the cell type which evolves to this normal plasma cells, so the early stages of the plasma cell differentiation. So basically these cells are also involved when you are vaccinated in generating new immune response, and also when you encounter an antigen again which you have previously encountered, like a measles virus or so, and these cells are then able to redifferentiate into plasma cells and to again produce the antibodies. So you cannot avoid that also the normal plasma cells are targeted. Mm-hmm. But as said, this is not critical, because they are reproduced again in the circulation. And this effect of removing normal plasma cells is also something one sees very regularly in myeloma patients, because as you know, there is also immune suppression in the myeloma patients. So when they produce, myeloma cells produce monoclonal antigen, then the normal protective immune globulin level also drops. And once myeloma is successfully treated, they also come back to normal levels. And this is exactly the same what we think will happen once the myeloma is treated with hopefully the TCB in this case.
Mm-hmm. So your body will regulate it essentially. So now I know that in patients there can be different levels of BCMA expression. So does this present a challenge for this type of treatment? So, and why does some patients have more BCMA than others?
So this is just a biological variable. So as there are men and women who are taller or less tall, that's just a physiological difference which appears from individual to individual. What you can say is that the level of BCMA expression in the myeloma cells is almost as high as in the normal plasma cells, so there's not a huge amount of variation in between.
So
and
this
is just a biological
BCMA,
thing, you know.
Yeah. Mm-hmm. Right. No, go ahead.
Okay, but the problem is the point is rather whether this is a problem for treatment, because let's say those patients who have less high expression on their myeloma cell surface would eventually react less well. But this is not the case, because we found that even with very low numbers of BCMA molecules on a myeloma cell, you still have a very good activity of the compound. So just to give you an idea, usually on one myeloma cell there are several thousands of BCMA molecules, about 2,000 in the median. And we even saw a killing of myeloma cells when there were only 42 in contrast to several thousand molecules on the surface. So it does not pose a clinical problem. And the reason for this is that the binding of this BCMA grabbing arm is really strong and is therefore able to grab the myeloma cells also when there's not so many of these BCMA antigens on the surface of the cell.
Well, that's fantastic that it's not an issue. So that's great. That's great news. So I have a question. Is BCMA expressed in MGUS and smoldering myeloma patients? So I guess at earlier stages of myeloma development or at earlier stages of plasma cell or B cell development.
Okay, I think Anja will take this question.
Yeah, this is again an interesting question also for patients. If you think of maybe treating also patients at earlier stages, and in fact we analyzed about 1,000 CD138 purified plasma cell samples, including patients with MGUS, but also smoldering myeloma, symptomatic myeloma, and relapsed myeloma, and we found BCMA to be expressed in all these stages. So there's no difference in the BCMA expression between the early stages and the late stages.
So it potentially could be used earlier, right?
Yes, of
if
course.
Approved.
This
that'd be
could
great.
be an option also for, let's say, high-risk smoldering myeloma patients. Yes, absolutely.
Oh, that's great. Well, that kind of brings me into another question about different genetics. As a lot of patients know, not all myeloma is the same, and every patient will have different genetic features on their myeloma, and some will have higher risk features and some will have kind of standard risk features. So does this discriminate between myeloma genetics, or probably not, because it's just going after the BCMA, right? It doesn't care if you have a deletion 13 or 17 or a 11-14 translocation or anything like that.
Yes, this is indeed the case. So we looked again also for this and looked for different risk stratifications, not only cytogenetics, but also gene expression-based risk scores or proliferation, and also tumor mass, for example, the ISS stage, also the revised ISS, and there was again no difference in the BCMA expression between, let's say, low or standard risk patients and high risk patients. So it's again applicable in principle to all patients, irrespective of the risk of the myeloma. Yeah.
Yeah, that's fantastic. That's great, because then you don't need a specific or personalized version, I guess. So tell us a little bit about what you've studied so far. In your study, how did it work for newly diagnosed or relapsed or even heavily pretreated patients? Because it seemed like reading your paper that it worked for everybody.
Yeah, actually, this is one of the few examples where myeloma is really easy, and this is one of those examples because indeed the compound acts in newly diagnosed patients, but also in relapsed patients. And in the manuscript, actually, we have also one patient who had several rounds of pretreatment, who was heavily pretreated, and we would not have suspected that the patient's myeloma cells would also respond, but this was actually the case, and they were almost cleared from the sample. And this is a specific surprise because usually we think that there is an immune defect or a suppression of immunity in our myeloma patients because they are prone to infections and so on. So you would rather think that also the T cells, having seen all this pretreatment and the exposure to anti-myeloma drugs and so on, are no more that active, but it's not the case. And this is, of course, very important because, as in all clinical trials, the first patient to be treated with the compound will be a relapsed patient. So, indeed, that's was very good news. And we looked in total for 48 patients, and it basically showed activity after 48 hours in over 80% of these patients, where you find a very significant reduction, and in some of the samples, all myeloma cells were killed in this, for myeloma, relatively short amount of time. So we are thinking that indeed it will work throughout the stages, as Anja had put for the genetics, also within the patients who are relapsing, and also patients at a situation where the treatment options might be very limited. Yeah.
Mm-hmm. Well, that brings up a couple questions. So I know I've heard other doctors talk about T-cell exhaustion, and where the T-cells just get overworked and then they get tired. Maybe that's why the myeloma cells grow out of control in the first place. But does this ever happen with the CD3 T-cell, or have you seen any of that, or is that totally irrelevant? Is this kind of a dumb question, which it could be?
No, I would not say, because this is what we were also confronted when we presented the data at the ASH meeting, for example, because this T-cell exhaustion is, of course, one of the things you are more or less afraid of when you give T-cell bispecific antibodies, but there was no indication that the cells could not be killed. And our explanation for us is, if we can think of this dog example, that these energetic or less active T-cells are a little bit like dogs who are prevented from biting by having this muzzle on their nose. And it really seems that this antibody, by bringing the cells together, allows this to be removed, and T cells being again active. And the evidence for this is given that many of the patients were heavily pretreated. So it's not at all a dumb question, but it seems that the antibody is clever enough to move away the muzzle from our T cells, and they are again able to attack the myeloma cells.
Mm-hmm. Okay, that's good. Wow, that's good news. Now, in your paper, you mentioned that a certain number of particular number of myeloma cells can be killed at one time by the CD3 T cell. So, if there's a high tumor burden or very fast-growing cells, what does this mean for patients who have a really heavy load of myeloma? Mm-hmm. Does it have any variety of impact?
This is an interesting question also. You have to take into account what fast-growing means. And you have to answer this question to set into perspective the doubling time of a myeloma cell, which is usually not below several weeks, and the time for killing of one myeloma cell, which is around this 6 to 8 hours. So even if myeloma grows fast, then still the killing of the myeloma cells is pretty much faster by a high amount, let's say 10 or 100 times faster compared to how long myeloma cells take to multiply, to divide and proliferate. And this means that it should also work in patients who have quite high tumor load and aggressive myeloma, because even then the doubling time, or the time in which the myeloma proliferates, is quite lower. We had seen, however, in our paper, in an animal model, in a mouse carrying a myeloma, that in 3 of 6 animals there was no activity. 3 of 9 animals there was no activity, or only a minor activity. But in this experiment there was used a myeloma cell line which had a very much higher proliferation or accumulation rate. So only in this setting of a mouse, we were not able to see some of the myelomas being removed. And this again is the rate of how the cell lines proliferate is much higher, so we don't see that this might be a problem in the myeloma patients, because we also saw, when you look at the number of T cells which are in our specific sample, that this can be quite low compared to the number of myeloma cells. So to summarize this, from what we know yet, this should not be a problem.
Mm-hmm. That's great news. So now I know that there have been some articles come out recently about daratumumab, that sometimes when patients start getting treated by daratumumab, their cells will start losing the CD38 signature on the surface, and so then daratumumab has nothing to go after. Can patients lose this BCMA signature also, or is that kind of escape unlikely?
Well, this is a very good question. Basically, there have been publications several years ago showing that there are no viable plasma cells without BCMA. So if you remove it in an experiment, then also the plasma cells die. This prompts us to think that it is a very good target, because one should think that the myeloma cells cannot downregulate it or completely cut it off, so that the grabbing of the TCB goes into void. But of course, this is something which in the end has to be tested within the clinical trials, because myeloma cells are, of course, quite clever when it comes to avoiding strategies, and this might eventually later on prompt not to use the compound as a single compound, but also in combination with other compounds where we might discuss on later on. So is there evidence that myeloma cells could lose a BCMA in our experiments, or for what is known in the literature? No, but you can of course not exclude it until you have really tested the compound within a clinical trial.
Mm-hmm. Yeah, that makes a lot of sense. So let's talk about how you use it in the clinic, or how you've used it in the clinical trial. So how is it administered? It's an infusion? And then how often, and what kind of schedule would you be using? Those types of things.
Yeah, so we have tested it in the animals, giving it IV or subcutaneously. Basically, it has a quite long half-life time in the animals of about 4 days, and presumably longer in humans, because in animals it's always shorter than in humans for this kind of antibodies or TCBs. So the idea would be to give it once or twice a week, either IV or either in the vein or subcutaneously. So it would be more or less comparable from the schedule to antibodies like daratumumab are given, but the exact way of how it is dosed and how often it is given to lead to the maximum effect in treatment would of course have to be determined within the clinical trials, in the phase 1 clinical trial. But this is the idea, at least this is the treatment schedule we used in the animal experiments to that end.
Mm-hmm. And then how often, I guess you'd have to test that in the clinical trial too, but what would your initial guess be if you had to say how often is administered? Is it something like daratumumab, where you get the first infusion, it's kind of longer, and then you get it repeatedly, but it kind of spaces out over time? Or I guess you would just have to see, right?
You cannot really answer this, because you have, of course, to balance there the convenience to the patient. This is the main reason why you decrease the daratumumab over time, because you think that you come into a saturation, so you cannot get too much higher when you add another round every week or every 2 weeks, and then it's also convenience for the patients. And for this, it's really too early to answer. But as said, the initial schedule will most likely be twice a week. But then everything has to come from how it really works out, and also from how the pharmacokinetics are in the patient. So meaning how long the compound is stable in the patient, how fast it is excreted, and so on. But most likely we will start with twice weekly administration.
Oh, okay. So let's talk a little bit about side effects or toxicity, infusion reactions, that kind of thing. I know with the CAR T-cell work on BCMA, and that's probably the most well-known BCMA type of target in myeloma, is that it has a cytokine release. So can you, in reading your paper, it sounded like this approach had a really low toxicity profile.
Yeah, that's absolutely true. I mean, also from the additional data we have obtained up to now, you see no severe toxicity, or you see no toxicity in terms of organ toxicity or anything like this. And the only thing you actually see is some mild cytokine release in the monkeys, which is not surprising because you more or less cannot activate T cells without some cytokine release, but it's clinically very, or it doesn't, in the animals, doesn't hinder them in any way. Of course, this is something you have to look at in the clinical trials, but we, of course, know how to administer antibodies because they're used for quite some time. So I would say that this is not really a problem. And definitely not something which would be more of a problem compared to other antibodies like daratumumab. So the really good thing is, as we see it now, is there's a very favorable toxicity profile for the drug, which also connects to a question you had early on, whether this might, and Anja nicely answered that, whether to treat patients also with, let's say, asymptomatic myeloma. That, of course, if it really will have this toxicity profile, this would be, of course, another argument to rather treat the patients early. But again, in the end, you only know when you conduct the clinical trial. So we have high hopes that it's not toxic, but we have to see in the patient, at least, however, from the preclinical data in the animals, there's nothing which prompts us to assume that there's some toxicity or strong toxicity.
That makes sense. So why don't you outline when you see this coming to a phase 1 clinical trial? How does that work in your regulatory process, and when do you estimate that might happen?
Well, actually, there's a quite set schedule to bring compound TCB compound anti-BCMA into the clinic, and this is scheduled for the first quarter of 2018. So this is really quite fast. You know, it always takes some time to develop a compound and then to do all the regulatory things. So this is presumably done, or it will be done, actually, I know, because I'm part of this, of course, within the year, and so that we will be able to start hopefully in the first quarter of 2018 with the enrollment of the first patient.
And how many patients will you have in that first trial? This will be obviously a phase 1 trial, right?
This still has the process of being determined with the authorities, which are involved quite early, because it's also a novel type of treatment, and we would like to go for innovative strategy in performing this trial. Usually the number of patients is anything between 20 and 80, and this has to be determined in detail. So we do not yet know, because this is still something in the process of discussing. But it's clearly not in a large trial including several hundred of patients, but rather to this number.
Mm-hmm. And I know in early phase 1 clinical trials, you're not really thinking you're just thinking, does this, you're trying to assess dose and safety and things like that. And then a phase 2 trial, you'll look at how effective it is. And once you get to phase 3 trials and beyond, then you start thinking about to combine it with other therapies. But in general, as a myeloma expert and team of experts, you've been working on myeloma for a long time, and typically myeloma therapies are combined. So could you see this being combined with other myeloma therapies? Which ones, and how would you see that happening?
I think this is the several million-dollar question to answer, also because all the compounds are quite costly. But besides that, I think we will have to determine what the efficacy of the compound really is in the phase 1-2 trial, which will be a combined phase 1-2 trial. But then clearly you would think of combining it with compounds which stimulate the immune system, like lenalidomide would be an obvious choice, and also it would be an obvious choice to use daratumumab, which attacks, as we have discussed initially.
Oh, I think okay, well, we lost our doctor, but they have directions to call back in, so we will do that and wait for them to call back in on the line. Sometimes, and they're calling in from Germany, so this happens on occasion, but we're not finished with our questions yet. So we will be watching for them. So they're back online. Hang on a second. Okay, sorry, we lost you for a minute. So go ahead, and you were answering that question about if combining with myeloma therapies. So apologize for the system dropping you.
No, so obviously I shouldn't have made the joke about the myeloma treatment cost. Don't know whether there's some connection. I presumably, of course, not. No, the combinations we were, there are all kinds of conspiracy theories, but I think we can exclude this here. No, so come back to the topic. I think there are some natural combinations, and one would be lenalidomide, something stimulating the immune system would, of course, make sense, and also it would be an idea to test the compound together with the daratumumab, for example, because the two compounds have both quite good toxicity profile, and they attack the myeloma cells from different directions. And obviously you would think also of combining it with something like dexamethasone, and this again has to be determined, but in the end we would also think that most likely it will be part of a combination therapy, which then again would be, if the hope holds true that it's really having a good efficacy but a low toxicity, then it would be a very good combination partner. But again, you never know until you have tried.
Right, and maybe the system just knew how much patients hate dexamethasone and dropped the line when you said it. I don't know. I think the idea of daratumumab or other immunotherapies would be so exciting. And I know that dexamethasone impacts the immune system, kind of suppresses it. So that would be interesting to see a drug like lenalidomide or daratumumab or this all, combined as a triplet or something. That would be really fascinating to see how that affects the immune system.
This is indeed a very interesting question, and there's also the discussion also within panels of myeloma experts whether it might be a good idea to add dexamethasone to antibody treatment, and also, of course, specifically to things like T-cell bispecific antibodies, and there are two effects which go in different directions. The one is that dexamethasone is one of the most active agents in killing myeloma cells, and the other is that you are always, of course, afraid that you hinder especially T-cell function. And which of the two would be more important, again, would have to be determined in a clinical trial. I would rather put my money on the dexamethasone, but as I do not have to take it, I can easily say this.
Load us up.
Again, you would just have to determine it, because there's no way to do this upfront without really seeing how it works out in the.
Well, completely. So one thing that I think is really interesting about this is that in a lot of the other immunotherapy approaches that are coming out right now, they're more personalized. So let's say the CAR T cell, you're modifying a particular patient's T cells. Is this more of an off-the-shelf approach that patients could get, similar to, an off-the-shelf drug that we have right now in myeloma?
Yeah, exactly. I mean, it's just like from the way you give it, let's say like daratumumab or things like lenalidomide, they do not, it's not in any way patient specific, which is the good point, because, and this is also why we put so much work in checking that it's really expressed on all the different, we had the genetics of myeloma and all the different stages and so on. So this is shown, and so it's really, or is foreseen really to be an off-the-shelf medication.
Right. Can you compare and contrast, I guess, with other immunology or immunological type of approaches? Like I've heard a little bit about the BITE technology, and then we are actually funding a CAR T-cell technology project. So I'm quite familiar, and I know a lot of people are more familiar with that. I know the BITE is kind of newer, but maybe you could kind of put these things in perspective for us.
I think the main advantage over the BITE technology is that with our antibody we have an extended elimination half-life, so allowing, as Dirk has pointed out already, a convenient dosing schedule. For the BITEs, in contrast, you usually it usually requires a continuous infusion via a pump that is carried by the patients for weeks, and which, at least to me, is maybe not that convenient. So this is, I would say, the main advantage over the BITE technology. Compared to CAR T cells, I think, as you have already mentioned, for CAR T cells you need a patient-specific generation, which is costly, and EM801 is, as we have already heard about, in principle applicable to all patients, and furthermore, the TCB antibody is, of course, eliminated from circulation within maybe around 1 to 2 months, compared to described long-term persistence of engineered T cells in case of CAR T cell approaches.
Mm-hmm. Well, it's so fascinating. I'm really thrilled that you are working on this. It's such an exciting opportunity, I think, and I think it's really genius that you have come up with this and are working on it. So I want to open it up just for caller questions. If anyone has a question, they can dial 347-637-2631 and then press 1 on your keypad. And we'll have a caller question at 310-5598 first. Go ahead with your question. Hello?
Hello.
Go ahead.
Doctor,
yes.
Thank you for joining our show. Your work is happening in Germany. I have a quick question. Could U.S. patients join a phase 1 clinical trial if they wanted to?
Sorry, I think I didn't hear it correctly.
I think the question is if you are running a clinical trial in Germany and a U.S. patient wants to join, is that possible?
Well, this is principally possible, but we will not only run the trial in Germany, but there will also be likely one site in the U.S. being opened. In principle, that would be possible, but of course, it would have to be negotiated with the respective insurance from the American side. From our side, it would not be a problem. And the plan is, and we also should mention this, that it's an initiative which is run not only by us, but also initially from the colleagues from EngMab, so the initial company producing it, and it will be also be opened in one site in Spain together with Bruno Paiva and Jesus San Miguel, who is the head of the group there. So it will be planned to be at 3 sites, So we also we also have had this in mind when choosing the site. So there will be very likely also open the site in the US. So you don't have to come to Germany for being recruited for the trial.
Okay, great. Thank you so much.
Okay. Thanks for your question. So let me ask you some more questions about EngMab. So would the clinical trial be a pharma-sponsored clinical trial, which would be open in multiple sites, like you're saying, in Germany and Spain, and then sites in the United States? Or is this, or how does that work? And maybe you want to share a little more about that company as well.
Okay, so this is also an interesting question. This company has started as a 3-people company by Klaus Strein and Professor Klaus Strein and Dr. Diem-Min Wu, and just one investor from Switzerland, Erich Hunzinger. Then they developed the antibody together with us, and as I mentioned, also with the clinic from Professor Jesus San Miguel from Pamplona in Spain and Bruno Paiva. And then we presented the data, or actually Anja presented the data at the ASH meeting in 2015. And then later on something really strange and funny happened, because then the company was bought by Celgene for actually a huge amount of money, so for 600 million euros.
Wow.
Which is more or less the same than US dollars. But before you ask, we did not get anything for this, so we still have to continue working, which we would anyway do. But then it was acquired by Celgene actually, and the trial will be sponsored and run by Celgene. But of course, we and also the initial EngMab team and also the group of Pamplona will be involved on this. And for the phase 1/2 trial, the plan at the moment is to have 1 or 2 sites at the respective countries, but also to start the trial obviously in the United States. So it's really a quite interesting story, almost, of course, for those who initially started it, a bit like a fairy tale. But it explains also why we are really proud to be part of this, and really looking forward to see the first patient treated. Because, this is our ultimate motivation, treating patients and seeing that at some time you might end in a situation which is very difficult for the patient because you do not have any more treatment options. And then on the other hand, of course, always this kind of research can be really frustrating when you try to develop compounds or work in this, and then at one or other stage it does not work out. But in this case, we are really pleased and happy that we will indeed bring it together with the colleagues to the clinical trial.
Well, to me, this is a great success story. I interviewed Dr. Craig Cruz, the inventor of carfilzomib, and he talked about this valley of death period that happens between that initial development that you talked about with, 3-person company. And it's a challenge because you need the funding in order to get the research results, in order to look exciting enough to be pursued. And this sounds like a terrific opportunity and one that was picked up quite successfully. So that is terrific that you will have the resources that you need to continue to run these clinical trials and take it all the way through the process. It's very exciting. One thing I want to mention is that this show was founded on the importance of joining clinical trials. And products like this or research like this that's happening is really moving the bar in myeloma therapy and helping you as researchers come to better conclusions more quickly. So I would just encourage patients to be aware of trials like these, and we will continue to follow your progress and post on this topic when the clinical trial opens, so that patients are aware of it and can join it. But it's really critical for patients to think about joining a clinical trial, whether they need treatment right now or not. In my opinion, you really need to plan ahead and be aware of things like this that are happening in the world of myeloma, so that if you need to make a decision on your treatment, you can put yourself in a position to join a clinical trial like this.
Oh, I think I could not agree more to this, because you have to be aware that everything we know now about myeloma and also which drugs work and which actually don't is based on former patients being included in clinical trial and being willing to help in this, even if several of these patients did not actually by themselves profit from eventually the treatment there. But only by this joint effort, as I mentioned initially, between physicians and also patients. And this, as you mentioned, makes it very important for patients to consider being part of a clinical trial. So only with this we will eventually be able to cure myeloma at one time. And you might also want to stress that there are, of course, very different types of clinical trials. So some trials where the medication is almost approved, or which are at a very late stage, so several hundreds or even thousands of patients have already been treated, and then in contrast those trials where it's really, as we have talked today, something which is the first use of a drug in men. And of course one has to stress to discuss with your treating physician as a patient what might be the options, and then to think about it. But the important thing, and in this again we could not agree more, it's important to think about whether to participate in a clinical trial. And this is really absolutely important. And to this end, we of course also thank all our patients who are willing to undergo the extra effort. Always you have to come some extra times to the center where the trial is performed, and we really appreciate also the effort of all our patients to be willing to undergo this, eventually to have, for a scientific question, then another bone marrow aspiration, which of course we also know as volunteers for this to be quite a painful procedure. But we really appreciate it, and I think if we will be able to cure myeloma, we will only be this as a joint team between physicians, researchers, and patients.
Well, I believe it is mutually beneficial. I think it's wonderful when patients decide to do that. I think they should always do that with their own care in mind. But I've seen research that shows that patients who join clinical trials actually do better too, with their overall survival. So I think, and maybe that's because they're followed so carefully. It is extra work and extra effort, but you really do benefit from it. Well, Dr. Hose and Dr. Seckinger, I am so appreciative that you joined us today. What an amazing topic, and what exciting research. I'm just thrilled that you're working on it, and I can't wait to see what happens in early 2018 with this study. Really, really pleased, and thank you so much for joining us today.
So thank you also for this opportunity to discuss about it, and also for your effort to really bring this to the patient. And I think shows like this bring together patients and you as an organization, and also us as clinicians and researchers, is a wonderful thing to indeed have a discussion and spread. So we thank you, and we'll begin our evening while you are only in the middle of your morning, perhaps for the post-show coffee eventually. At least this would be what we would do. And so we wish you a very good day and all the best from Germany.
Well, thank you so much for joining us. And to our listeners, thank you for listening to Myeloma Crowd Radio. Tune in next time to learn more about the latest in myeloma research and what it means for you.
With Lucky Land Slots, you can get lucky just about anywhere.
Dearly beloved, we are gathered here today to... Has anyone seen the bride and groom?
Sorry, we're here. We were getting lucky in the limo and we lost track of time.
No, Lucky Land Casino, with cash prizes that add up quicker than a guest registry.
In that case, I pronounce you lucky.
Play for free at LuckyLandSlots.com. Daily bonuses are waiting. No purchase necessary. Void where prohibited by law. 18+. Terms and conditions apply. See website for details.